Good day, this is Professor Will, CBMD PhD. Today's Monday, December 7th, 2020. It is 6:20 and 55 seconds a.m. Eastern Standard Time. And this is a continuation of my microbiological research, particularly genetic risk factors. So let's hop to it. Strategies for boosting accuracy of personal genetic risk scores. Date December 4th, 2020. Source Scripps Research Institute. Summary. As the consumer genetics industry rapidly expands, more and more people are turning to DNA-based services to learn their risk of developing a wide range of diseases. However, the risk scores from these genetic tests are not always as precise as they could be, according to a new study. The scientists examine many approaches to calculating the scores and recommend that personal genomics organizations adopt standards that will raise the bar for accuracy. Polygenic risk scores can be incredibly useful and affordable tools to guide preventative health decisions. Given the increasing utility of these scores, it is important to remove variability as much as possible and ensure the data is updated regularly to reflect new knowledge from genomic studies. Companies such as 23andMe and Ancestry, along with dozens of others, need only a small sample of person saliva to generate estimations known as polygenic risk scores, which are determined by comparing snippets of an individual's DNA with findings from large-scale genome studies. Examining these small segments of genomic variation, known as SNPs, is faster and far more economical than sequencing a person's entire genome. But one drawback is that the results can sometimes vary unexpectedly. Researchers note that in most cases, the degree of fluctuations in a risk score is small and doesn't change the overall interpretation of the result. Most individuals remain in the same risk category. Also, fluctuations are especially minor at the extremes of the risk distribution, where disease risk implications are most critical. Yet, any variation can lead to a loss of consumer confidence in these important scores, and that can undermine preventative health. And in some rare cases, the scores can fluctuate substantially. For further study, a research team used various methods of calculating polygenic risk scores for conditions including coronary artery disease, atrial fibrillation, type 2 diabetes, Alzheimer's disease, glaucoma, and breast cancer. They found that regardless of the method used, the computational algorithms are prone to introduce random variability for individuals due to how the calculations incorporate data from population-level genetic studies. To reduce variability in individual polygenic risk scores, the team recommends running the algorithms multiple times, which would help smooth out random imperfections. By paying attention to variable elements in the score calculation, actions can be taken to either eliminate those elements or create an average. While researchers identified some clear challenges in applying population genetics to individual-level genetic analysis, researchers also see ways to overcome these issues to produce results that will inspire confidence. Researchers decided to pursue the study after making updates to a different project they developed called MyGeneRank, which predicts a risk of heart disease and other conditions by looking at specific genetic markers. They sought to identify a computational process that would lead to the greatest degree of stability and accuracy in the face of a score that is expected to evolve over time as new data becomes available. The recommendations put forth in the new study could help personal genomics companies improve the quality of the scores they deliver to customers, and ultimately encourage more individuals to take smart preventative health actions based on what they learn in genotyping reports. Good day, this is Professor Will, CBMD PhD, on microbiology, computer science, and bioinformatics. Well, hope you enjoyed listening. Until next time, have a great day.